So this lecture is a brief introduction to phage biology. We'll talk about what bacteriophages are and a little bit about their bacterial hosts. So you can find all these slides on Canvas, but I'm going to break this lecture into two shorter videos, one focusing on the phages and the other focusing on the bacteria. So for each unit, you need to pay attention to the learning objectives. So learning objectives are things that you should be able to do by the time you get to the quiz. So some things for this unit: you should be able to explain the differences between a virus and a cellular organism, describe the basic structure of a bacteriophage, diagram the reproductive cycles of bacteriophages in detail, explain why lytic and lysogenic reproductive cycles are advantageous in different contexts, diagram the basic structure of a bacteria, calculate changes in bacteria number during growth and culture, explain the concept of viral host specificity, define the bold terms in slides, and write scientific species names using correct formatting. So if you're not sure how to do some of these things by the time that we get to the end of the lecture, make sure you ask your instructor for some more details. The homework will give you some practice in doing many of these things. So what is a bacteriophage? So bacteriophages, or phages for short, there are viruses that infect bacteria. You can see on the right-hand side of this slide, there is a transmission electron micrograph that shows a number of bacteriophages. So here on the outside of what is a bacteria cell. These phages are infecting the cell by attaching to it and injecting their DNA into the cell. You can actually see that on the inside of the cell, some of these bacteriophages have begun to reproduce. So they are making new viruses inside the bacteria. Bacteriophages are the most abundant biological entity on the planet. There are an estimated 10 to the 31st bacteriophages on the planet, and that is a 1 with 31 zeros on the end, just to give you an idea of how many phages and how big that number is. There's a graph on the bottom that shows some big numbers in context of the number of phages. So viruses are a type of biological entity that many people don't consider to be alive. So there's some differences between viruses and the things that we traditionally think of as living organisms. So you recall from biology that living organisms are made of cells. So the cell is the basic building block of organisms. Some organisms, like us, are made up of many cells that work together, and some organisms, like bacteria, are made up of individual cells. But all cells have genomes made of a molecule called DNA, and that DNA holds all the genes, and so those are the things that have the instructions for making the cell work. And the DNA allows those cells to produce all of the components that cells need to carry out the processes of life. They have DNA replication machinery that lets them make new copies of their DNA. They are able to make proteins from the genes in that DNA. They're able to use those things to get energy from their environment and then to reproduce on their own. So viruses have some similarities and some differences from living cellular organisms. So they're not cellular. They are instead composed of a protein capsid, so it's a hard shell instead of the soft, oily cell membrane that surrounds most cells. And there's often other molecules attached to that capsid. They have genomes that are made of DNA, but they can also, some viruses have genomes that are made of RNA, which is a similar but different molecule. The biggest thing that makes viruses different is that they can't reproduce on their own. They don't have all the components that cells do to replicate DNA, to make proteins, and to get energy from the environment. They need to hijack a host cell in order to reproduce. So most bacteriophages are DNA viruses, and the DNA viruses that are bacteriophages are in the viral order called Caudovirales. They are tailed bacteriophages, so they have a capsid attached to a long tail, and The genomes of these phages are made up of double-stranded DNA. There are other types of bacteriophages. There are RNA phages, such as the phage Q beta or in MS2, but most of what you'll find in this class are caudovirales. Those caudovirales fit into 3 viral families, so the myoviridae, here on the left-hand side. So those are phages with a long contractile tail, and that tail can contract like a syringe to inject the DNA into the bacteria. In the middle here, these short stubby ones, these are podoviridae. And then the siphoviridae, here on the right-hand side, so they have a long flexible tail. So what do these tailed bacteriophages look like? So this is a diagram of one particular tailed phage called T4 bacteriophage. So bacteriophages have 2 different components. They have protein components and they have DNA components. The protein makes up most of the phage, so it makes up the capsid and also the tail and the things that are attached to the tail. And then the DNA genetic materials usually stored within the capsid. This is a 3-dimensional picture of that phage, and so you can see the protein capsid shell and the long tail, and also these tail fibers that allow the bacteriophage to attach to its bacteria host. So this is a model of T4 bacteriophage, but on the right there are some transmission electron micrographs of some other real phages. So this is corn dog, which has a longer capsid up at the top and a long flexible tail. Caliche, which has a more rounded capsid, but also a long flexible tail. Lidotin, which is a myoviridae, so it's got this capsid up here, and then it's got the contractile shorter tail. And then burrow, which is a podoviridae, it's got just this little stubby tail attached to that big capsid. So, like we said, phages need to infect bacteria in order to reproduce. And so how does that happen? So T4 bacteriophage has to attach to a cell and then inject its DNA into the cell. So why do these bacteriophages do this? Well, phage capsids, tails, and spikes are all made of protein. But phages don't have any of the machinery to make protein on their own. The phage's DNA genome has all the instructions for making all these phage proteins. But in order to make those proteins, the phages have to get the bacteria to make new proteins. And so they do that in a process that we call the lytic reproductive cycle. Right? So a bacteriophage, in order to start the cycle, has to attach to a host bacteria and then injects through that bacteria cell membrane and cell wall its DNA. So once that DNA is inside the bacteria, the DNA circularizes so that it looks somewhat like the bacteria's own DNA genome, which is shown here. Once it's inside of the cell, that bacteriophage then gets the cell to replicate the phage DNA. So now we have lots of phage genomes inside of the cell. And that phage DNA has all the genes, all the genetic instructions for making new heads and tails and other phage proteins. And so the host cell makes all of these components inside of the cell. Once they're all made, all of these particles can assemble inside of the cell, and then the DNA genomes get packaged inside of the capsid. So now we have a whole bunch more viruses, and so they secrete an enzyme that breaks open the cell in a process we call cell lysis. And that releases these phages in the environment, so they can go find a new bacteria host to start this process over again. Right? So each time a phage infects a bacteria, it reproduces inside the bacteria and produces dozens or hundreds of thousands of new virions, and then it kills that cell, and then they go out and find new cells to infect. Right? So what is that process going to look like to you? Because all of these things are too small to see with the naked eye. Right? So What we'll use is an assay called the plaque assay. So we'll talk about this in detail when we talk about bacteria, but this is what a plaque assay looks like. We have a petri dish with some bacteria on it. So that bacteria looks like this gray growth that's all over the plate. We call this yellow-gray opaque looking bacteria growth turbidity. So when those bacteriophages lyse the bacteria, it kills those cells and it clears an area of turbidity on the plate. Right? So when those phages infect a bacteria, they reproduce and kill that bacteria, make a bunch of new phages, and those phages spread out and kill bacteria in a surrounding area on this plate. And so that creates a little circle where all of the dead bacteria form what's called a plaque. Right, so this is an area where there's no turbidity, there's clearing on the plate. So the lytic cycle is a really effective way for bacteriophages to reproduce. By infecting a single bacteria, they can produce thousands of new phages, and then those phages can all go find new bacterial hosts, infect those phages, kill the bacteria, make thousands more phages, right? But the problem with this is that the bacteria die in the process, right? So imagine a scenario where those bacteriophages fall into a small pool and there's just a handful of bacteria, maybe 100 different bacteria in that pool. And those bacteriophages infected the bacteria, produced that bacteriophage infected one bacteria, produced thousands of new phages, and then that bacteria was lysed and killed. And then those thousands of new phages infected the other 99 bacteria in that pool. And they all produced thousands of new phages and killed all those bacteria. But now there aren't any more bacteria left in the pool. And even though there might be thousands and thousands of phages at this point, what are they going to do? They can't go anywhere. They can't relocate themselves. They don't have arms or legs or ways to get around in their environment. They're stuck. And eventually, if they don't find some new bacteria hosts, they'll die. So some bacteriophages have come up with a different strategy. Instead of tricking the bacteria into making thousands of copies and then shredding the bacteria to bits, those bacteria are able to reproduce in a different way. So lysogenic reproduction is an alternative to the lytic cycle. So as we talked about in the lytic reproduction, bacteriophage infects a single bacteria, injects its DNA into the phage, and then reproduces new phages which kill the cell and release those phages into the environment. But a lysogenic reproduction looks like this. So instead of producing new phages, that DNA genome gets cut and pasted into the bacteria's genome. So it integrates, it cuts and pastes itself into the bacterial DNA. So that genetic material for the phage, right here, with all the instructions for making phage genes, is now part of the bacterial DNA. So bacteriophages that can reproduce through the lysogenic cycle are called temperate phages. So temperate phage means that they can reproduce both lytically and by the lysogenic cycle. So this is how the temperate phage works. So the phage infects the cell, injects its DNA genome into the bacteria, and that viral DNA integrates. It becomes part of the bacterial genome. And so now those bacteria cells have all the machinery needed to produce new cells, and so they grow and divide, And every time they copy their DNA and split into two new cells, the phage genes here, they double in numbers. And now there's two copies of the phage genome where there used to be just one. Every time the cell divides, two new copies of the phage genome. And every time the cell divides, the phage genome doubles in numbers. Right. However, under stressful conditions, the prophage, which is what we call the phage genome while it's in the bacteria, right? So these little bits of phage DNA in the bacteria are the prophage. The bacteria gets stressed. We'll talk about what some things that are stressful for bacteria are. The prophage DNA gets excised. It gets cut out of the bacterial chromosome, and then it enters the lytic cycle. And we call this process induction. And then, once it's in the lytic cycle, those phage genomes are turned into proteins. The phage capsids and tails and all the parts of the phage, they get assembled into new viruses, and then those viruses lyse the cell and go find a new host. Right? So why do the temperate phages reproduce as a prophage in some cases, and then as a lytic phage in other cases? Right? So stressful conditions which induce this lytic reproduction for temperate phages, stressful conditions are anything that makes it hard for the bacteria to grow. I can't write very well with this tool apparently. However, stress is anything that makes it hard for the bacteria to grow. So really extreme heat, lack of nutrients, ultraviolet radiation that can damage the bacteria's DNA. All of these things tell the phage that this bacteria isn't going to go through the reproductive process anymore, and it's time to go find a new host. So that is what induces the prophage to come out of the DNA, make new phages, so those phages can go find a bacteria that's maybe doing a little bit better than the one they're in right now. Right. So this is a diagram of temperate phage reproduction. Remember that temperate phages can reproduce either as a lysogen, right, as part of the bacteriophage, or they can reproduce lytically. Right? So while they're a lysogen, right, there are things that prevent the lysogen, prevent those lytic genes from damaging the cell until stressful conditions induce lytic reproduction. So it's going to reproduce as a lysogen inside of the bacteria as long as it can, and only reproduce lytically when it needs to. So why do we study phages? So one big reason that we are studying phages now is that phages have been shown to be an alternative to treating antibiotic-resistant bacterial infections. So we'll talk about a story of Dr. Stephanie Strothy, and she wrote a book called The Perfect Predator and has a TEDx talk. So Dr. Strothy, her husband got infected with a actinobacteria infection that was going to kill him. These were some bacteria that infected her husband that were resistant to every single antibiotic drug that we have on the market. And Dr. Strothy assembled a team that was able to discover some bacteriophages that were able to treat and kill the bacterial infection that her husband had, and they were able to save his life. In the middle here, so Dr. Paul Turner is a scientist at Yale, and he is developing phages that actually force bacteria to lose antibiotic resistance. He's got an iBiology series on YouTube that has some really good information about viral ecology and phage therapy. Finally, some of the phages that have been found in the C-Phages program, so that's the program that phage hunting is part of have actually been used also as phage therapy. So just the other year, Isabel Carnell Holloway, so she was a 17-year-old in the United Kingdom. She was actually had her antibiotic-resistant infection cured by phages found by C. Phages students, and we'll talk about that later in the course. But we're also going to study phages because they're really neat. Right? Many of the important discoveries in biology came from studying phages. We'll talk about Hershey and Chase. They discovered that DNA is the genetic material by using phages. Many of the other important discoveries, such as that mRNA is the intermediate between DNA and protein, came from phage experiments. And most recently, a number of scientists, including Jennifer Doudna and Emmanuel Charpentier, discovered a new enzyme called CRISPR, which is part of a bacterial defense system that bacteria use to prevent phage infections. We now use CRISPR to edit DNA in humans, and it's been used recently as a therapy to cure a number of chronic genetic conditions. So we don't always know what discoveries are going to be important in science. So basic or curiosity-driven science is critical for uncovering these new possibilities.